Hello guys and welcome back to Excel Academy. So in previous lectures we talked about the DNA polymerase and how exactly the initiation of the replication is being started, right? So what are the important points? Can anyone summarize? Like what is the first thing in the initiation of replication one should be considering? What is the first step? Shruti, can you tell the first thing?
The ORI and binding of DNA protein A, which has to recognize ORI and create the duplex so that replication can start from there.
Where you have to mention about the 5.
The 5 9-mer sequences have to be identified by DNA A to bind.
Yes, very good. And when we have got the 13-mer, 3 sequences are there where the first duplex will be formed, where the AT-rich region is present and the strand separation will start. Very good. So that's how you have to talk about the initiation. Okay. So now we'll be talking about the elongation. Here nothing much is there because once the replication has been started, the DNA polymerase will just keep on adding the new nucleotides. Okay. So the synthesis of the 2 new daughter strands takes place complementary to the template strands, and DNA polymerase are the enzymes. We have got 3 types of polymerase in the case of prokaryotes. That is DNA polymerase 1, 2, and 3. One is responsible for filling the gaps, and like where in the previous lecture we saw where the RNA primer is removed. So the gap is filled by the DNA polymerase 1. 2 is responsible for the repair mechanism. Whenever DNA undergoes repair, any damage is there. So that time DNA polymerase 2 is going to function. And the third one is the holoenzyme. This is your main enzyme which is carrying out the replication. So DNA polymerase can synthesize in one direction only, which means that they can add DNA nucleotide, that is in the 5' to 3' direction. So whether it is prokaryotes or eukaryote, the DNA replication or DNA synthesis is always in 5' to 3' direction. Now, of course, the DNA polymerase require primer, and who is providing the primer? What is the name of the enzyme? Yes, Tanvi, what is the name of the enzyme which provides the primer? Yes, very good, primase. And that is also known as what kind of protein in prokaryotes?
DNA G.
Yes, it is DNA G. Very good. So primer is around 5 to 10 RNA nucleotide sequence, which is specifically required for the starting the new strand of DNA. Now, this DNA polymerase can extend this RNA primer, adding the nucleotide one by one, and for all this process, it requires ATP. So, every time, whenever a nucleotide is being added, ATP is required. Now, we know that there are 2 types of strands, leading strand as well as the lagging strand, right? So, leading strand. So, this runs from 5' to 3' direction towards the replication fork. जैसे जैसे आपका replication fork जाएगा, उसी direction में आपका leading भी जाएगा. So, if suppose replication fork is going in this direction, आपका first जो इस तरफ जा रहा है, वो आपका leading रहेगा. उसके opposite में आपका lagging रहेगा. ठीक है? So, for leading, you just need a single primer to start the extension action of the DNA pol 3. Now, lagging strand, we know again it is 5' to 3' direction, but away from the replication fork. In this only we have got the Okazaki fragments, and this requires more than one primer. It is a discontinuous process. Now, why it is called discontinuous process? Because we know every small stretch of RNA primer is being synthesized. जितना ज़्यादा आपके पास primer होगा, उतना ज़्यादा small stretch of DNA will be synthesized. Okay. Now, once this RNA is being removed, now this RNA is being removed with the help of dash enzyme. What is the name of the enzyme? Which enzyme removes the RNA primer?
Ma'am, RNase H.
Yes, very good. It is RNase H. Okay. So it can remove all the ribonucleotide, but only RNA and DNA hybrid जो रहता है वो कौन remove करेगा? Yes. RNase H can remove only the ribonucleotide bonds. It cannot remove the hybrid bond which is formed between the ribonucleotide and the deoxyribonucleotide. ये कौन remove करेगा?
Ma'am, exonuclease.
कौन सा exonuclease?
5'
DNA polymerase.
हाँ, okay. Now, don't get confused. It is the 5' exonuclease activity of DNA pol 1. ठीक है? Pol 3 has got 3' exonuclease activity, which we have discussed when we were talking about the how the incoming nucleotide sits in the binding pocket and कोई error हुआ, mismatch हुआ. So the proofreading activity of the DNA polymerase. So DNA polymerase 3 has got 3' exonuclease activity. 5' exonuclease activity is present in DNA pol 1. Okay, I hope abhi clear hai. There is no doubt. Now last step is about the termination of replication. So we know that replication of bacterial genome, it is bidirectional and terminates at a position diametrically opposite to the origin of replication. So हमने figure में भी देखा था last time that if you have got यहाँ पे origin, ठीक उसके diameter में you will have the terminator sequences for the termination. ठीक है, अगर आपका origin यहाँ है तो ठीक उसके opposite में diametrically opposite में यहाँ पे आपका termination sequence होगा. Okay, now here also if you remember the replication fork is moving in this direction and in this direction. So eventually what will happen, both are going to meet over here. The replication fork is going to meet at this termination sequences. ठीक है, अभी हमें ये देखना है how exactly these terminating sequences are stopping the replication fork. नहीं तो क्या होगा, you could also imagine that if suppose these sequences are not there, what will happen? There's a chance that replication fork can go ahead, right? And यहाँ पे जो replication हो चुका है इसको override करेगा, उसको दोबारा वो replicate करेगा और यहाँ वाला यहाँ को जाके करेगा. So what will happen? There won't be any stopping of the replication. It will just keep on replicating. Eventually what will happen? Just imagine a bangle which is having so many bangles, ऐसे. So this is how the genome of the bacteria will look like if there is no stopping. ठीक है, इतना clear हुआ आप लोग को? The point which I want to convey, is the message clear to everyone?
Yes, ma'am.
Huh? Okay. Now, so so replication terminates at a terminus region containing multiple copies of 20 base pair sequences called ter sequences or the terminus sequences. Now this has got the protein which goes and bind. It's called TUS. That is terminus utilization substance protein. So अभी क्या है? You have got the bacterial genome. यहाँ पे origin है. यहाँ पे ter sequence है to which your TUS protein binds. Okay, so if suppose this is your ter sequences, यहाँ पे you are going to have proteins which are bound to the ter sequences and something like this. They are facing like this. So for example, अगर replication fork यहाँ से आ रहा है, it will just go and because of this TUS protein, this replication fork will stop of this direction. अब वो इसको override नहीं करेगा, ठीक है? अगर आपका रेप्लिकेशन फॉर्क इस तरफ से आ रहा है, so because of this facing TUS protein, it is going to stop here. So this is not going to pass like this. Stop हो जाएगा. थोड़ा सा आइडिया मिला? So TUR is the sequences to which these two are your TUS proteins. ठीक है? Now, so E. coli DNA termination sites, they have got A, B, C, D, F, G are found in the DNA. Of these, TUR A terminates the counterclockwise moving fork, while TUR C terminates the clockwise moving fork. Now look at the figure over here. So you have got the origin. उसके ठीक opposite में, diametrically opposite में, you have got the TUR sequences. Okay, these are the TUR sequences. Now, when you have got the counterclockwise fork coming, okay, so which is going to stop? Can you see it over here? Direction देखो यहाँ पे, TUR C, TUR B, TUR F, TUR G का direction इस तरफ है, ऐसा बना हुआ, right? TUR A, D and B का direction इस तरफ है. Can you see the differences in their structure?
Yes, ma'am.
So what you have to remember is over here, TUR A at the end over here, TUR A is going to stop the replication fork for the clockwise fork, whereas TUR C is going to stop for the counterclockwise fork. अब हर एक रेफरेंस बुक में आपका जो स्ट्रक्चर है ऑफ द TUS प्रोटीन, इट इज गोइंग टू बी डिफरेंट. ठीक है, यहाँ पे ऐसा दिया है सीक्वेंस और कहीं और पे कुछ ऐसा दिया होगा. जस्ट अंडरस्टैंड दैट दिस, अगर ये आपका इस फॉर्म में दिया है तो ठीक उसके ऑपोजिट में ये स्ट्रक्चर दिया होगा. जस्ट अंडरस्टैंड दैट मच. ठीक है. Look at over here. See again, different structures they have got shown. So यहाँ पे क्या होगा? Now the TUS protein will come and bind. Again you can see C and A, the direction. ये कुछ इस तरह से फेस कर रहा है, यहाँ पे ये इस तरह फेस कर रहा है, right? So now you have got the TUS protein blue color का, which comes and binds over here. Okay, so now this is called TUR-TUS complex. This is important, guys. So origin हमने देख लिया. So when you talk about origin, you have to talk about ORIC. वहाँ पे 13 mer and 9 mer के बारे में पढ़ना है. You have to talk about the DNA binding proteins. ठीक है. Then you can talk. That is the initiation of your replication. Elongation में कुछ ज़्यादा नहीं था. After that you have got the termination का. In termination you have to talk about the TUR sequences, and then you have to talk about the TUS proteins. Okay. So इसलिए ये important हो जाता है. Now again same thing, हाँ, एक ही चीज समझाने के लिए. So you have got the TUS protein. So you can let us say that this is for your TUR C and this is your TUR A. Okay. So you can see the directions ऐसे and इस तरफ दिया हुआ है. Now what will happen? The replication fork can pass the TUS protein bound in one direction. Now जैसे ही replication fork यहाँ से pass होगा, what will happen? You can see there is a structural change in this TUS protein, and after that, after passing this TUS protein, it is not able to pass the second one because the conformation or the orientation of the second TUS protein is different, and that is what is blocking the replication fork to move ahead. Similarly, अगर replication fork इस तरफ से आता, so this TUS protein might have gone under the conformational change, और इसका structure कुछ ऐसा हो जाता, and then because of the difference in this structure, तब ये आपका structure ऐसा रहता. So replication fork is not able to, or we can say that this TUS protein is blocking the replication fork from this direction. So अभी क्या हो गया? Finally यहाँ पे आपका replication fork खत्म हुआ. अब दूसरा आएगा, दूसरा भी आपका यहाँ पे आके खत्म हो जाएगा. Getting the point, guys? Is it clear to everyone?
Yes,
Ma'am, the first one में it passed through, right?
हाँ, in both the cases it
The
is going
second one
to
also
pass
would.
through. In both the cases it is going to pass. See,
So at the
जैसे
last one
पास
there will
हुआ,
be some DNA.
नहीं
Yeah.
सुनो बताती हूँ. ये भी जैसे पास हुआ, you can see there is a conformational change, right? There is a conformational change, and because of this conformational change, replication fork is not able to go ahead, and it is blocked by this protein. ठीक है? Okay? तो यानी कि ये replication fork पहले पहुंच गया है to the termination site.
Okay.
Okay, it is not like this. One second. It is not like this कि exactly both the replication will be reaching the end point at the same time. ऐसा नहीं होता है. Okay, यहाँ से शुरू तो हुआ, but the time taken by this replication fork and this replication fork going to vary. ऐसा नहीं है कि ये 5 मिनट लिया है तो ये भी 5 मिनट लेगा. Okay, so
no, but it wasn't about the time. The second this protein over here, even the DNA would also pass through that, right? So you'll end up with this middle portion where there may be double multiplied DNA or something like that.
हाँ, DNA replication नहीं समझ में आया श्रुति. Like, what is your doubt actually? See, this protein is present over here. This is also double-stranded DNA. This is also double-stranded DNA. हाँ.
Yes. So look, in the first case, the sequence passed through. It continued multiplying until it reached the second Ritas protein, right? And that is because of the difference in conformation, it couldn't pass through the second one. Right? So same thing should apply to the other, from the other side when it's coming through. Replication technically should be passing through this one and stopping at the next sequence because there's a difference in conformation, right? So you'll end up with a place in the middle where there are multiple, where it has been replicated twice.
कैसे श्रुति? See, first of all, don't say multiplication, say replication. हाँ, सब गलत जाएगा meaning. हाँ. So see, this is double-stranded DNA. Now, what about this? Even this is also double-stranded DNA, है ना?
Yes. Yeah.
है ना? Okay.
Yes.
Now it is reaching over here, but it is not able to pass through. What does it mean? It means the DNA ball has fallen off from here. गिर गया है. अब नहीं है वहां पे. ठीक है? Now what is happening is from this side, replication
other
fork
side
is
the
coming.
DNA ball is coming in.
Yes, it is coming.
Yeah.
It is passing, and just because यहाँ पे ऐसा खुला रखा है, it is coming. ये भी तो ऐसे आ रहा है ना? Finally, it is going to merge.
Okay.
Huh? It needs to merge.
Yeah, I thought it would go into one of them because there was no.
No. It is going to merge. Otherwise, क्या होगा? हमेशा termination sequence में फिर we won't have the what to say, the daughter strand getting, right?
Yeah.
तो करना ही पड़ेगा. So it depends whether this replication fork is crossing and stopping or this replication fork coming and crossing and stopping. At the end, I want two double-stranded DNA. है ना? एक parental हो जाएगा और एक आपका daughter strand का हो जाएगा. So at the end, I'm gonna get this concatemer. Okay, tangled wala. Then you know that you are going to have the topoisomerase coming and helping or gyrase coming and helping. ठीक है? So this is what you get at the end. So this catenation problem is coming. So you are going to have both. So finally, the topoisomerase or the gyrase is going to come, make a cut, double-stranded cut, open up the DNA, bring it out, and reseal it. Okay. So this is how you get two copies of the DNA, and finally, then the multiplication of the bacteria or the division of bacteria is happening. Okay. Now, ये थोड़ा important है because MCQs comes from here only, the 2 marks card. ठीक है? Not the entire, but you have to remember about the DNA A, B, C, and E, G. G भी ठीक है. G तक ही आपको पढ़ना है. ये N, Q, T नहीं पढ़ा तो चलेगा. Then you should know about the gyrase A and B. This is your ligase. ओराई पॉल आपको मालूम ही है. देन RNA is H. इसका locus है. Locus means where the gene is present. ठीक है. Then you have got the single stranded binding proteins TER and TUS. Clear? 2-3 बार इसको रीड करोगे, finally you will remember also. No problem. Now there's one thing about beta unit of Pol 3 enzyme. Okay, that maybe in the evening lecture we'll talk about it because it's a big topic and it is important one also. Okay, so this is upon you guys. आपको ये पूरा टेबल अगर आप याद कर सकते हो तो अच्छी बात है. Otherwise, at least those what we have already discussed. ठीक है. Phenotype की जरूरत नहीं है. Locus and the function. If that much has been understood, that is more than enough. Okay, now coming to the specialization of DNA polymerase, this is nothing, just already we have discussed like a DNA poly 1, 2, 3 क्या करता है. So the central role of DNA polymerase in the it is the efficient and accurate replication of the genome, which has resulted in the evolution of multiple specialized DNA polymerase. Now guys, who can explain me this sentence? इस sentence का मतलब क्या हुआ? Yeah, anyone? Tanvi, can you explain me this sentence meaning? Yes. Anyone else? Tanvi, Akshaya, Chandni? Shruti?
So since DNA polymerases are very important, but they also have a fairly diversified portfolio, as in like they have a fairly diverse role to play. So they have evolved and differentiated to specialize in certain functions, which finally contribute to the same function of DNA synthesis. So each DNA polymerase has evolved in such a way that it can maximally do one particular job very efficiently.
And since our DNA is so big, सिर्फ एक DNA polymerase से करना मुश्किल है. ठीक है. So it is not possible for having all the responsibility given to the one particular enzyme. So the DNA polymerase, they have evolved and got specialized. Like एक repair के लिए चला जाएगा, एक छोटे इस काम के लिए चला जाएगा, एक बड़े काम के लिए चला जाएगा. So this is how they have evolved, just to maintain the efficiency of replication without any error. ठीक है. Eventually we'll see that there are many errors even after so much proofreading and so much care has been taken, but फिर भी mutations होते हैं. We'll talk about that in the next topic. Anyways, okay, so that is the correct answer. So they have got at least 5 DNA polymerase, which that they are distinguished by their enzymatic properties, subunit composition, and abundance, of which 1, 2, 3 we have already discussed, and 4 and 5 भी आपका repair में आता है. ठीक है. So DNA Pol 3 is a main enzyme for the replication. Since the entire 4.6 MB E. coli genome is replicated only by 2 replication fork, so this has to be highly processive, है ना? Because यहाँ पे सिर्फ 2 ही replication fork है. There is only one origin of replication, so it has to be very, very efficient. It is not like the case of the eukaryotes where you have got thousands of origins and thousands and multiples of replication fork working over there. Okay, so it has to be highly processive. So DNA polymerase 3 is actually a part of a larger complex, which is very having the high processivity, and it is known as DNA polymerase 3 hollow enzyme. So this hollow enzyme means it has got the sliding clamp, beta clamp, all these are coming together to form the hollow enzyme, a part of which is DNA polymerase 3, which is carrying out the replication. So agar aapko previous figure yaad hoga, so there we have seen that on the template, okay, on the DNA you have got such a big molecule of the beta clamp or clamp loader hum bolte hain. theek hai. So it is holding like this, having here one DNA polymerase and here another DNA polymerase, jahan se aapka lagging strand aata hai, ek aapka leading strand pass karta hai. So you have got the loader over there. So those are, then you have got the beta clamp, sliding clamp, all these coming together to form a hollow enzyme. Okay. Now, in contrast to the polymerase 3, we have got DNA pol 1 also, which is specialized for the removal of RNA primers. और जहाँ पे भी gap होगा, it is going to fill with the new nucleotides. It also has got 5' hexanucleus activity because it is responsible for removing the DNA RNA का hybrid जो present रहता है, ठीक है? DNA pol 1 is not highly processive. It can add only 20 to 100 nucleotides per binding event, and that much is enough, right? Because हमारा primer भी इतना ही होता है. So after removal, this gap can be easily filled by the DNA pol 1. Okay, and now this also we have discussed RNase H के बारे में. Okay, again same thing. Okay, so pol 1, pol 2, pol 3. So here you can see pol 3 core हो गया है hollow enzyme. It is responsible for the main chromosome replication, whereas rest of the polymerase are involved in the DNA repair. So this TLS, UMUC, D2, ये सब हम तब पढ़ेंगे when we'll talk about the mechanism by which the polymerase are helping in the repair of the DNA. ठीक है? Okay, now this is the MCQ time. So few questions only I have put. Okay, don't worry, it's not that tough, it's easy only, and all these questions are from the previous year's question paper only. The only thing is I have not put the kaun se saal ka hai, 2016 hai ki 18 hai, whatever it is. So guys, are you ready?
Yes, ma'am.
Okay, so your first question is थोड़ा सा light हो गया है. I'll just read for you guys. So while replicating DNA, the rate of misincorporation of DNA polymerase is 1 in 10 raised to 5 nucleotides. However, the actual error rate in the replicated DNA is 1 in 10 raised to 9 nucleotides incorporated. Now this is achieved mainly due to dash. That is first option, spontaneous excision of misincorporated nucleotides. Second one, 3' to 5' proofreading activity of DNA polymerase. Termination of DNA polymerase at misincorporated sites. And last one, 5' 3' proofreading activity. Yes, so what will be the answer? Huh?
Option A, ma'am. Spontaneous excision of misincorporated nucleotides.
Okay, so Chandni says option A. What about others?
Option B, ma'am.
Yeah, option B.
Saying B, Chandni saying A, and what about Shruti?
B.
What is your answer? Okay, so the correct answer is B. Why? Because till now I taught you about the 3' 5' proofreading activity of DNA polymerase, right? And Chandni, there is never a spontaneous excision of misincorporated nucleotide. It is always regulated either by the DNA pol 3 हो गया या 1 हो गया. Okay, so you can't have spontaneous excision. You can have spontaneous mutation, because of which there will be a wrong incorporation of nucleotides, but you cannot have spontaneous elimination of the nucleotides. Randomly in the nature doesn't happen during the replication. Okay, so of course it is not the termination, and 5' 3' proofreading activity bhi nahi hai. So the answer is 3' 5' proofreading activity of the DNA polymerase. So this is also we talked about, right? When we were talking about the replication, मैंने mention किया था. See, जैसे ही कहीं पे भी wrong incorporation हो जाएगा, so your DNA polymerase, this is the structure they have shown over there. If you remember, Chandni, so we have got the double helical, and जहाँ पे भी mistake होता है, so there will be a bend, right? There will be a bend because the wrong nucleotide has been added. because of which what will happen? The DNA polymerase activity will reduce and its exonuclease activity will increase. So that exonuclease activity will do what? That is nothing but the 3'5' exonuclease activity. It is going to remove that wrong nucleotide which has been added. And once this has been removed, then again the polymerase is going to the activity will regain and it will start adding the new nucleotides. Is it clear now, everyone? Chandni, your doubt is clear?
Yes, ma'am. Yes.
So the correct answer is second. Good. So what is the number of hydrogen bonds in a double helical bDNA structure of 100 base pairs with 20 adenine and 10 thymine in one of the two strands? Okay, तो बहुत simple है. So कुछ समझ में ना आए, just draw the diagram. In one of the two strands बोला है, so you can take any strand. It has got 20 adenine and 10 thymine. Okay. आगे क्या करना है? Yes. What will be the next step? Yes. Akshaya, क्या करना चाहिए next?
So if there is 20 adenine and 10 thymine, then totally 30 and rest out of 100 base pairs. So 70 is GC pairs. And next, so we have to calculate the hydrogen bonds. For 30 adenine and thymine, so there are 2 hydrogen bonds. So 30 into 2, 60. And next, for GC bonds, 70 into 3, so which is 210. So 270.
Everyone agrees with this? So according to...
Yes, ma'am.
Yes, ma'am.
Good. So the correct answer is C. So I hope now everyone got the idea. आपके ऐसे भी question आए, you guys are able to calculate the hydrogen bonds, right?
Yes, ma'am.
Good.
Yes, ma'am.
So I hope you all got the confidence in doing this, right? अभी अच्छा हो गया है. So let it be percentage or whatever number. This is how you should do. ठीक है? Good. So the correct answer is 270. Next question. So during DNA replication, events at the replication fork require different types of enzymes having specialized functions, except DNA replication में कौन सा enzyme हमें नहीं चाहिए? Options are DNA polymerase 3, gyrase, ligase, and glycosylase.
D.
Ma'am, D.
Okay, everyone is saying the option D, that is glycosylase. So what is the function of glycosylase, guys? Glycosylase का function क्या है?
Maybe addition of sugar or something.
No. So DNA glycosylase are the enzymes which is responsible for repair, DNA repair. ठीक है? For example, by mistake, अगर uracil add हो गया है, so then DNA glycosylase is responsible to remove the uracil. It's a glycosylase, right? So what it does is that it is going to break the bond between the deoxyribose and the base, uracil base. तो क्या होगा? आपका base हट जाएगा, and what is remaining is deoxyribose sugar and the phosphate is remaining without any base, right? So then later the other enzymes will come and remove that without having any base का apurinic या apyrimidic जो हम बोलते हैं, so that will be removed. Anyway, so we'll talk about it in detail in the upcoming lectures. So the answer is D over here. The DNA glycosylase is not required during the replication. It is meant for the repair. Okay, good. Now, origin of replication usually contains dash. ये भी आपको आना चाहिए. This is like 1,000 times I have said in the class.
D, ma'am.
Yes.
Ma'am, D.
Correct answer is 80 rich sequences. Good. Now, in bacteria, chromosome DNA replication starts at dash. What is the answer then? इतने धीरे से A, are you not confident? The correct answer is A, one specific locus. As I mentioned, there is only one origin in the case of bacteria. Okay, so only one origin, so that's why it is only one specific locus. In the case of eukaryotes, you have to tell that it is several specific locus. It can never be random. Whether it is bacteria or eukaryotes, it can never be random. ठीक है, so bacteria case में हो गया आपका one specific locus, eukaryote case में हो गया several specific loci. Okay, good. Next question. In semi-conservative mode of DNA replication, and I'm guys telling you, जितना बड़ा question होगा, उतना ही simple answer होगा, ठीक है? You should be afraid of short questions. जो big questions होते हैं वो ऐसे ही होते हैं time pass के लिए, but they are very easy. In semi-conservative mode of DNA replication, two parental strands unwind and are used for synthesis of new strands following the rule of complementary base pairing. So you know what is semi-conservative, right? हमेशा एक parental रहेगा और उसके ऊपर daughter strand आएगा. So that is semi-conservative. Synthesis of complementary strands require that DNA synthesis proceeds in opposite direction, while the double helix is progressively unwinding and replicating in only one direction. One of the DNA strand is continuously synthesized in the same direction as the advancing replication fork, which is known as the leading strand, whereas the other one is synthesized discontinuously in segments and is known as lagging strand. अब तक वही बोल रहे थे जो हमें already पता है. So these short fragments are known as Okazaki fragments. This also we know. So these Okazaki fragments need to be matured into continuous DNA strand by which one of the following combination of enzymes? Yes.
B, ma'am.
B. Give me the answer. मुझे B, D में थोड़ा confusion हो जाता है सुनने में.
DNA polymerase 1 and DNA ligase.
Okay. What about others?
Same.
Okay. Are you sure it's the DNA pol 1 and DNA ligase and not DNA pol 3 and DNA ligase?
No, ma'am. DNA pol 3 is the one that works on the leading strand. It is more efficient at adding a lot more nucleotides compared to pol 1.
DNA polymerase 3 extends.
The thing is, DNA pol
Yeah.
3 is working on both the strands, leading and lagging. Okay, it's just that Okazaki
No, I meant
fragment.
Okazaki
fragment.
You have to get in between the base pairs to fix the Okazaki fragments. Fragments, and DNA pol 3 does not do that. It extends.
Yes.
DNA pol 1 goes in between and fixes the gaps.
It fills the gaps when RNA primer is removed and the nick is present. Okay, so DNA pol 1 is coming in between, filling the gap, and ligase, of course, sealing the आपका DNA strand. Good. Okay, so that's all. So how was MCQ? Was it difficult? Was it difficult? These are like from previous year's question. So अगर यही question अगर दोबारा आते हैं, I'm sure everyone can do. Right? सिर्फ एक question में एक को confusion हुआ था. Rest all you guys were able to give the answer straight away. So it's a good.